All right, we're here to talk about photosynthesis today, so let's get started with this process. Photosynthesis is what we call an anabolic process, meaning that this is something that's going to be putting molecules together instead of breaking them down. So we're going to put things together, and anytime we put things together, we're storing energy. So that's really the goal here. Photosynthesis is only carried out by plants and some bacteria that have the organelles necessary to do that, which in our case is going to be the chloroplast that we're going to be talking about. If you look at our reaction for photosynthesis, we see carbon dioxide, water, energy on the reactant side, and that's going to produce carbohydrate and oxygen. Well, that's basically the exact opposite of what we saw for cellular respiration. And that's basically how it works. The plants make the energy, they get the light energy from the sun, they convert that into chemical energy that everything else on Earth can use. And this is kind of like the circle of life going on, is that they make the energy and then they can break it down because they have mitochondria and they have the organelles where they can use this they make it for themselves. For us to get the energy, we either have to eat the plant or eat something that ate the plant. And this is kind of that circle of life kind of thing that goes on when we talk about biology. Photosynthesis happens in two different pathways. There's two basic steps that are going to be happening. We have the light reaction or the light dependent reaction because it needs light to take place. And we have the carbon fixation reaction, which has a couple other names. We could call it light independent because this can happen without light at all. Some people will call it the dark reaction because, again, it doesn't take light. Or you may hear the name Calvin cycle, which guess who figured out that this was going on? Yeah, it was Dr. Calvin. So names it after himself like pretty much every scientist does anytime they discover something kind of cool. There are some prokaryotes that can do things with photosynthesis, but for the most part, when we talk photosynthesis, we're going to be talking about plants and the reaction that's happening within the chloroplast. So when we look at the chloroplast, we're going to see a double membrane structure, very similar to how the mitochondria works. Mitochondria had double membrane, chloroplast has a double membrane. When we look inside the chloroplast, there is the stroma, which is a liquid inside the chloroplast, kind of similar to the matrix inside the mitochondria. And then we have these little disc-shaped things that are stacked up all over the place inside the chloroplast. Each of these individual discs is called a thylakoid. That thylakoid, the stack of thylakoids, is what we're going to call the granum. So we have our outer membrane, we have our inner membrane, we have the stroma as the liquid around here, and then the discs are actually called thylakoids, and a stack of that disc is called the granum. So if we're going to talk about photosynthesis, we have to know a little bit about photochemistry. Photochemistry means that the closer the light source is to an object, the more intense it's going to be. When we look at the intensity, for every distance, if we have distance 1 here, the brightness is 1. At distance 2, it's 1/4. So basically what happens is we have something called the inverse square law, where if I put every time I increase the distance, you put that distance on the bottom of a fraction and square it, and that's how much less intensity there is compared to your original distance of 1. And that just basically means if you had a light that was 6 inches in front of your eyes, it would be about 4 times stronger than a light that's 12 inches in front of your eyes. It would look that much brighter, and that's because as light leaves the source and comes towards the object it's going to hit, it spreads out and it goes over a larger surface area, so it's going to naturally get a little bit weaker over space. When we talk about light, light comes in the form of electromagnetic radiation, and electromagnetic radiation can come in longer waves or shorter waves, just depending on what the light source is or what color you're looking at is going to be determined by these waves. Longer waves tend to have less energy than shorter waves. Now, when light hits an object, there's going to be 3 things that can happen. One of 3 things can happen. 1, it gets deflected or it's scattered. It hits off something and bounces off. If you ever shined a laser or a flashlight at a mirror, you see that the laser bounces off it. It can also pass right through it. Think about X-rays or electromagnetic radiation, and when they hit your body, they go right through you, just because they're that powerful. The other thing that can happen, and this kind of happens all the time lights hit something, is that the energy gets absorbed. And when the energy is absorbed, well, we say that molecule is at a ground state before the light hits it. When it absorbs energy from the light, it moves to what we call an excited state. And that basically means that the internal energy of that object sort of gets a little bit higher, it gets a little bit more excited. And that's what we're going to be talking about with the chloroplast, is how things get excited and lose electrons. We look at the ultraviolet spectrum, or the electromagnetic spectrum, sorry, that's the word I was looking for. We see that we have shorter wavelengths way up here. Remember, X-rays, those things that go right through you, really high power beams of electromagnetic energy, which means they're going to have a lot shorter waves that we see way up here. When you look at like a microwave, like you cook food with, look how long those waves are. Really not very hazardous at all. I mean, they're just really long waves that go there and they vibrate water molecules inside your food, then that's what causes the heat to happen. If we look in the middle of the scale, this is where visible light is going to sit. And we look at violet. If we got all the colors of the rainbow, Roy G. Biv right here. Red, orange, yellow, green, blue, violet. Violet's going to be one of the shorter wave guys, and red's going to be one of the longer wave guys. So red's going to be the lowest energy out of all the visible light, whereas violet's going to be kind of the highest energy of all the visible light. And these are all the different colors and sort of where they fit on this scale. This 400, 500, 600, 700 refers to how the distance between the peaks of all the waves. And again, this is kind of a little physics lesson, but that's okay. We have to understand a little bit of physics to understand how light works. When you look at something, right now I'm looking at my shirt and I have a blue shirt on. Well, basically what that means is that the fabric of my shirt absorbs every color in that visible light spectrum except for the one that I see. The color that I see is the one that's reflected back to my eyes, and my brain takes that wavelength and interprets that into a color, and that's sort of what we're going at. So if there's something that's red, that object's going to absorb every color except for red. When you look at plants, what color are plants usually? Usually those are going to be green. So if the plants are green, if that's what you perceive, well, what does that mean? That means that that plant is absorbing every other wavelength of light except for the green wavelengths. And that's going to be important as we move forward and when you guys do a lab on photosynthesis, which we'll be doing probably next week sometime. Chlorophyll is a compound inside the plant's chloroplast. And it is a molecule that is going to be really good at absorbing light. Chlorophyll comes in two forms. We have chlorophyll A and chlorophyll B, and when chlorophyll is exposed to light, it's going to absorb it. And as it absorbs that light, it gets really excited. That excitement happens because these things are absorbing. We talked about the electromagnetic spectrum and the three things that can happen. Light can either bounce off of it, go through it, or get absorbed by it. Chlorophyll is going to absorb the light, and when it gets excited, it's going to lose an electron. Once it loses that electron, it returns to that ground state or that normal area. So when we look at chlorophyll, that energy in chlorophyll gets absorbed, and the chlorophylls are set up inside the thylakoid of the plant. Well, the chlorophylls are all kind of connected in this kind of antenna system. And as the light comes in from the outside source, which is usually going to be the sun, or it could be a grow light, something like that, that'll provide the energy for the plants to convert it to sugars. Well, the chlorophyll absorbs it, sends that energy into a central chlorophyll, and they get transferred. And as these chlorophylls keep absorbing energy, they get excited and they lose electrons. Our photosystem is where all these chlorophylls are, and usually it's embedded in proteins that are sort of in the thylakoid membrane. There is the reaction center, which is where one main chlorophyll is going to stay, and that chlorophyll at the reaction center is going to absorb all the energy that the other chlorophylls absorb. And as it absorbs all that energy, chlorophyll is going to lose an electron. So loss of electron is oxidation. So that's what we see here. The electron is given up to an electron acceptor, which means the chlorophyll is going to have to get reduced, because remember, we can't have something get oxidized and then not get reduced pretty soon afterwards. So we have things going back and forth. We have redox reactions going back and forth. And think about cellular respiration. Cellular respiration, there's a lot of redox reactions happening. Same thing's going to go on in this process. We're going to have those redox reactions happening, which is going to help with the transfer of energy to make that actual carbohydrate. So when we talk about these photosystems, with the photosystems, that electron is going to get passed down an electron transport chain, very similar to that electron transport chain that happened in the mitochondria during cellular respiration. Guess what happens as electrons go down the electron transport chain? ATP gets produced, and the plant's going to need that ATP because that's going to fuel the processes later to help make carbohydrates. So let's look at what's going on with the photosystems. We have two photosystems when we're talking about photosynthesis. Now, don't think photosystem 1 has to happen before photosystem 2 because it doesn't completely work like that. We're actually going to start talking about photosystem 2 first, but that's okay. We'll just sort of get there in a second. We look at those two different chlorophylls in the reaction center of each of these photosystems. In photosystem 1, you have a P700 chlorophyll. That P700 is going to absorb light best at 700 nanometers, and that's that distance from peak to peak that we talked about on the electromagnetic spectrum. Photosystem 2 is going to have chlorophyll. That's what we call a P680, which, guess what? It absorbs light the best at 680 nanometers from peak to peak. So the shorter wavelength is going to be absorbed best in photosystem 2. The longer wavelength is going to be absorbed best in photosystem 1. So this picture is a little bit overwhelming. Let's just sort of orient ourselves so we know what's going on. Right up here it says photosystem 2. There's my P680 right there. This one on the right says photosystem 1. There's my P700 right here. These little wavy lines, these yellow lines coming from outside are going to be photons. Photons are code words for electromagnetic energy or light. As the photons come into the chlorophyll, they stimulate all of these different chlorophylls that are sitting up in this photosystem. You see these little antennas sort of going down here. That's all sending its energy down to that reaction center, or that P680. Well, as this thing gets excited, it's going to lose an electron. Now, it doesn't jump up and out of it. This is just sort of a picture that shows, hey, we have a high energy electron that just came out of here. Now, if it's lost an electron, this chlorophyll just got oxidized. If it got oxidized, it's going to need to get reduced very soon. Look over here just to the right. We have water here. If you remember our photosynthesis equation, light plus water plus carbon dioxide is going to produce glucose and oxygen. Well, here's water getting reduced into oxygen. If you remember from cellular respiration, this went the other way. Oxygen was an electron acceptor and eventually got converted into water at the end of that electron transport chain. In photosynthesis, the water breaks down and turns into oxygen and a couple hydrogen ions. Those electrons from the water breaking down are going to reduce this chlorophyll that's in photosystem 2. Now, I know I said, don't worry about the order of photosystem 1, photosystem 2. That doesn't matter. We're sort of starting with number 2 because it sort of fills in some blanks for us. Now, if we remember, this chlorophyll got excited, it lost its electron. That electron is going to cruise down the electron transport chain. And remember what happens during the electron transport chain? There's some chemiosmosis that causes hydrogen ions to leave the thylakoid, and they're going to go through ATP synthase. ATP synthase is going to turn ADP and phosphate into ATP. So we're starting to create a little bit of ATP here that's going to be used way later. Now, as this electron gets to the end of the electron transport chain, it needs something to accept it. Well, in photosystem 1, we had the P700 chlorophyll at the reaction center. Photons of light is striking this one too, and all of these other chlorophylls sitting on the outside of the reaction center. They send their energy to the P700 chlorophyll. It loses an electron. This one just got oxidized. It lost an electron. Guess what? That electron from photosystem 2 is going to reduce that chlorophyll and bring it back down to the ground state. This electron from photosystem 1 is going to go through an electron carrier, and that electron carrier is going to convert NADP plus and hydrogen into NADPH. Similarly, our body, when we did cell respiration, we broke down NADH into NAD+. You'll notice in photosynthesis, it's NADP+, not just NAD+. Similar molecule, just slightly different, the same basic processes. So we sort of have this cycle where photosystem 2 feeds photosystem 1. Photosystem 1 creates NADPH. This NADP+ is going to act as the electron acceptor in this instance. So this is sort of the words of what I just said. The excited chlorophyll, that P680, is going to give up its electron. That electron is going to go through the electron transport chain. As the electron goes through the electron transport chain, energy gets stored as ATP through chemiosmosis going through the ATP synthase. And in order to replenish this chlorophyll with an electron, water got oxidized, and that's going to reduce the chlorophyll. The water turned into the oxygen, which was a waste product for the plant, which, thank you plants, remember, what does oxygen do for us? That gives us an advantage because we can use oxygen to go through oxidative phosphorylation and cellular respiration. So that's kind of a big deal with that water getting oxidized. So let's look at that again. Same exact picture, same exact stuff. Photosystem 2, all the photons of energy get transferred to P680. It releases its electron. That electron is going to go down the electron transport chain. Since this chlorophyll lost an electron, water is going to get oxidized. And water is going to get converted to oxygen and hydrogen ions. Those electrons are going to refill the chlorophyll in photosystem 2. After that electron gets through the electron transport chain, it makes some ATP as there's some diffusion of hydrogen ions through the ATP synthase. This P700 is getting stimulated by the photons of light coming from the sun. It loses its electron because of that stimulation. That electron gets replaced by the one from photosystem 2. The electron lost in photosystem 1 is going to go through an electron carrier, and it's going to finally be accepted by NADP+ to create NADPH. Again, this will be used in later processes of photosynthesis. So now we sort of have a cycle and an end product going on over here. Here's the words to what I just said. The chlorophyll, the P700 that's in photosystem 1, releases its electron. The oxidized chlorophyll gets reduced by the electron from photosystem 2. Think about that as the link. That's what's connecting photosystem 2 to photosystem 1. Those electrons pass through and eventually reduce the NADP+ and NADPH. Now we have some stored products and good stuff is going on. Here it is again. I'll let you look at it for a second. I won't even say a word. Just think about what's going on. There we go. Think about the things I was just saying. The chlorophyll got excited. It released its electron. That electron has to get replaced. In photosystem 2, the electron is replaced by the oxidation of water. In photosystem 1, the electron in the chlorophyll is replaced by the electron from photosystem 2. A little ATP is created, and NADPH is created after photosystem 1. That's kind of the big stuff going on right here. Now, is photosynthesis over yet? Not exactly. We still don't have a carbohydrate yet, and that was kind of the big deal with photosynthesis, is that the plant's going to make glucose. The plant makes glucose so it can use that for energy for basic life processes. So we need some more reactions to make this happen. Now, in order to make glucose, we actually kind of need a lot of ATP. We need more ATP than NADPH, to be honest. So there has to be some sort of way for that to happen. That can happen through something called cyclic electron transport. Cyclic electron transport happens in photosystem 1 only, and it doesn't happen all the time. It just happens to create some extra ATP so that the plant cell has enough action to basically get through the Calvin cycle that's going to be coming up. Now, with cyclic electron transport, that P700 from photosystem 1 loses the electron. It goes through electron carriers and a separate electron transport chain from the one that we were talking about earlier. That's going to create ATP. This electron actually gets recycled and actually fills in the chlorophyll that it came from. So it just sort of drops it up, and it's just a big cycle, just like the name says, cyclic electron transport. The nice thing about this is that the plant can create ATP without creating any extra NADPH, which it doesn't need as much of. It's going to need a lot of ATP when we look at that Calvin cycle. So how do the gases exchange in the plants? We talked about oxygen was one of the waste products. Well, on the bottom of the plant, of the leaves are kind of like little solar panels, and they kind of turn towards the sun. If you ever had a plant that's sitting in a window, you'll notice that the stem will actually turn towards the sun so that the surface of the leaves are really exposed. On the bottom of the leaves, they have some special cells in an area called our stomata, and the stomata is kind of like two lips that kind of open and close. These vacuoles will fill with more or less water depending on how much photosynthesis needs to be taking place and open this pore up bigger or smaller. That's going to allow the carbon dioxide to go into the plant and the oxygen and water vapor to exit the plant over the course of time. Now we're going to talk about making that carbohydrate. Now, we talked about that stomata opening and closing to allow gas exchange. Oxygen comes out and carbon dioxide, is going to come in. About 6 molecules of carbon dioxide are going to come in, and they're going to come and go through this Calvin cycle. So I'm going to tell you the same thing I told you now before we looked at the Krebs cycle, because this is kind of like the plant's version of the Krebs cycle that it does during photosynthesis. Don't try to memorize everything on this chart. Let's go over the big thing. First thing we have to do is understand that most carbon fixation occurs when it's light outside inside the stroma. So just to sort of orient ourselves, our stroma is that liquid that's inside the chloroplast and is sitting around the thylakoids. Calvin cycle has 3 basic parts that happen. First thing that's going to happen is carbon dioxide is going to get fixated. It's going to get turned into a molecule that's useful for the plant. Once it gets fixated, there's going to be a product called 3PG is going to get reduced to form G3P. Now, I understand 3PG, G3P sound very similar. Sometimes I get confused. Just sort of focus with me and we'll get there. The last thing that's going to happen is that we need another carbon dioxide acceptor, which is going to be RUBP. And again, I don't care if you know the name for RUBP, I'll go over it in the slides, but if you can know RUBP, will be very, very more than good. But we need that acceptor of CO2, RUBP, because it's going to take the CO2 into the plant and sort of accept it and allow the carbon to get fixated inside the stroma. Now, this is a really important word right here. Rubisco is going to catalyze the reaction of carbon dioxide and RUBP. Rubisco, is probably the most abundant enzyme on the face of the earth. This enzyme is in every plant, and there's a lot of it in every plant because plants need to capture that CO2. They can't capture the CO2 without this Rubisco combining the carbon dioxide and the RUBP. When those two things combine in the presence of Rubisco, our product is 3PG. That is huge, huge stuff right there. Every time a carbon dioxide goes through the Calvin cycle, more carbon dioxide is fixed to an acceptor and RUBP is regenerated. As the RUBP gets regenerated, it allows for more carbon dioxide to go through this reaction. This Rubisco is extremely important in making that happen. Without that, the plants wouldn't survive, and if the plants die, guess what? We're not going to be too far behind them because they create all the energy that is available on the earth. Without them, we're kind of screwed, so we need that Rubisco to stay where it needs to go. It takes 6 molecules of carbon dioxide to get 1 molecule of glucose, and hopefully that makes sense because hopefully you know that glucose's chemical formula is C6H12O6. Carbon dioxide is CO2. So if I have 1 carbon here and 6 carbons in here, I got to go through it 6 times. So I need 6 molecules of carbon dioxide to go through that Calvin cycle. And here it is. Carbon dioxide comes in. Look at just to the left of that. The carbon dioxide has the arrow going to the right. RUBP is coming in from over there, and it accepts that 6CO2. I'd have Rubisco right here as sort of the presence, and that's going to create something called 3PG. Remember how I said we needed all that ATP? Look how much ATP is going in here. 12 molecules of ATP going in there are going to break down and create some bonds. Anytime we create bonds, we're creating places where energy are going, and that's the whole point of creating glucose, is that glucose is our preferred source of energy. The plant's going to use the glucose to run the plant's cellular processes. We're going to eat the plant or eat something that ate the plant to get the benefits of that glucose and have energy for ourselves. And as we go through there, the 3PG turns into G3P. You get enough G3Ps together, that's going to create a sugar. This circle goes around and around and around, and as it keeps going, carbon keeps getting accepted, and eventually you get that sugar to come out, and that is the huge, huge stuff going on here. Once G3P gets to the bottom of the chart, as I showed you on the previous slide, it's one of 2 options. It gets exported into carbohydrate, or later in the day, when it's a little bit darker out, the plant's going to store that glucose. How do plants store glucose? You guys remember that? Yeah, that starch word right there is a plant stored form of glucose. What is an animal stored form of glucose? We store it as glycogen. So the plant's going to store it as starch, and that starch is going to be in the plant for whenever it needs an extra sort of bankroll of glucose so that it can create energy. Now, why is this so important? Because this powers everything that goes on in Earth. Without the plants, nothing else happens in Earth. I mean, you really don't think about it, but the oil that powers your car and lights your house was the fossils of dead plants. Dead plants that lived hundreds of millions of years ago died, they got buried in the earth, and they got crushed. And that glucose and that starch that was in those things got converted into long carbon chains, and those large long carbon chains are energy. When you eat something, you're taking that glucose that's stored inside those plants. When you eat a potato, when you eat corn, when you eat rice, you're eating that stuff and taking that glucose and that starch and putting inside of your body so you can convert that into chemical energy for yourself through cellular respiration. These processes go hand in hand. When we talk about organisms that do photosynthesis, we call them autotrophs, which means self-feeders because they make their own food, they make their own glucose, or we also call them producers because they produce all the energy that is left on the earth. Those autotrophs and producers have provided food for other things. And when we get to ecology, we'll talk about the food pyramid and all that, and how you got producers, first order heterotrophs, second order, third order. But hetero means other, trophs means feeders, so other feeders. They have to feed on other things, and we are all other feeders because we're animals. No animals can produce their own glucose. We're just not that cool. So we need to eat other things in order to get our energy, which is another reason we are called consumers. We have to consume things. And that's sort of the basis of photosynthesis and what's going on there. Well, I hope you enjoyed that. It was a long one, but you know what? That's stuff that we need to know and stuff that we're going to be looking at over the next couple days in class, and something that we'll be looking back at when we do our lab on photosynthesis. Hope you enjoyed, and I will talk to you soon.